Okay. Let's get restarted because I have a lot to get through. So let's do it. So as I was saying before, having parental genotypes as controls is ideal, but as we all realize, the real world doesn't conform to what we'd like in many ways. And one of those ways is there's a lot of missing data. I mean, everyone in theory has a biological mother and father, so in theory you could always do this. But in practice, rarely are those genotypes available. More common type of data is sibling pairs, as I was saying before. Also pair and offspring pairs being collected for many kinds of data, especially often mother-child pairs. So one potential solution of this missing data problem, where parental genotypes are not available, is to impute the missing parental genotypes based on the observed genotypes according to the laws of Mendelian inheritance. And this is the basis of this paper that we have in review at the moment. It was an idea I developed with my postdoc supervisor, Augie Kong, originally. It's been going on for a while now and involving quite a lot of people in the SSGAC, including some of the research assistants, and especially for developing this software that's available on GitHub now, Sniper, so that does the imputation and also the genotype-phenotype association analyses for individual SNPs and for polygenic indices. So I actually have a computer exercise that I'm going to put in the Dropbox that is optional. It's not necessarily part of the course, but if you're interested, there's a computer exercise that goes through a sort of tutorial on how to impute missing parental genotypes and also some of the other stuff I'm going to talk about. So just to give an idea of how this works, this imputation, basically, if you have a sibling, this is imputation from a sibling pair. If you have a sibling pair, two siblings either inherit the same or different alleles from their father and the same or different alleles from their mother. So they can share 0, 1, or 2 alleles by descent from their parents. And that's what I'm showing on the top here. They can be an IBD 0, which means they inherited different alleles from their father, different alleles from their mother, and in which case you've observed all 4 parental alleles. If they're in IBD state 1, then they've inherited the same allele from the father and different alleles from the mother. And you've observed 3 parental alleles, and one of them is unknown, and you can impute the unknown one with the population frequency of the allele. If they're in IBD state 2, that means they've inherited the same allele from the father, the same allele from their mother, and there are 2 unknown alleles. And these different states occur with different probabilities depending on the laws of Mendelian inheritance. You can derive the probabilities. IBD 0 state happens with probability 1/4. IBD 2 state also happens for probability 1/4. And IBD 1 state happens with probability 1/2. So that means by doing this imputation, inferring which alleles of the parents you've observed in the sibling pair, you can recover 3 out of 4 parental alleles on average, because you recovered 4 here with probability 1/4, 3 here with probability 1/2, and 2 here with probability 1/4. So you can actually get 3/4 of what you're missing just from a sibling pair. And if you have more siblings, you're going to recover more on average. So that's a sibling pair. Pair and offspring pair, in many ways, is more trivial. And there's a slight technical aside that it's not really worth going into, but it is important that you need phase data to really do this as well as you can. But in this case, where the mother is heterozygous and the offspring is homozygous, you can see that, well, the offspring must have inherited an A allele from the mother, and it must have inherited its other A allele from the father, so you can impute 1/2 of the father's alleles trivially, except in the case where both parent and offspring are heterozygous, in which case you can't determine which one was inherited from the mother and the father, unless you have phase data, in which case you can use the haplotypes, not just the individual SNP to do this. So that's what we do in practice, but in principle, it's trivial when you don't have this double heterozygous case. So you can recover 1/2 basically of the missing parents' alleles when you have a parent-offspring pair. And obviously there are lots of other data types possible. You can have multiple siblings and one parent genotyped, or both parents genotyped. You don't need to do any imputation, but basically you can derive a sort of efficient imputation of the missing parents' alleles based on the observed parents by applying the laws of inheritance to figure out which parental alleles have been observed, potentially using phased haplotype information too. And so what you're doing in this case, so actually I haven't shown this model before, but this is just a slightly more general way of doing the family-based GWAS than what I showed before. You only need the sum of the parents' genotypes to get unbiased estimates of the direct effects, but you might want to allow the coefficients to differ on the paternal and maternal genotypes, so that's what I'm showing here. And so you're fitting this multiple regression model, and let's say that you've observed the individual's genotype and the mother's genotype, and you've imputed the father's genotype as missing. You've imputed it based on the individual and the mother's genotype. What you're essentially doing is imputing the missing father's genotype as a conditional expectation given the observed genotypes, i.e. the individual and their mother's genotypes. And then you're thinking, oh, okay, can I do a multiple regression where I'm replacing the true unobserved paternal genotype with the imputed genotype? Is that going to give me a sensible answer? It's not obvious that it would, because you don't have the true unobserved you don't have the full model. This is only an incomplete reconstruction, essentially, of the missing father's genotype. But it turns out, and this is kind of cool, that if you, in general, actually, you can prove this, that replacing unobserved covariates in a multiple regression with a conditional expectation given the other covariates, as long as the resulting regression design matrix is not collinear, you essentially recover consistent and unbiased estimates of the fully observed model parameters, along with valid sampling errors when you do this kind of imputation. And the reason that it works in this case is because we're basically imputing the missing genotype as a nonlinear function of the observed genotypes. It's nonlinear because Mendelian inheritance is like a digital process. And because it's nonlinear, you gain information in a multiple regression context. If it was a linear imputation of the unobserved genotype, you would have a fully collinear design matrix and you wouldn't be able to do the regression. So because Mendelian inheritance is a digital process, it's not like a linear and whatever kind of combination of things, averaging things, then you can gain information for this multiple regression without adding noise. And that's why you get these nice statistical properties that are pretty unusual. Imputation is a big area in statistics, and usually imputation methods don't have these kind of nice properties when there's a substantial amount of missing data. So we're seeing how the unique properties of sort of biological genetic data can actually lead to a sort of statistical method that has interesting properties.
So that what will be off, though, is the variance of the imputed genotypes?
So the variance of the imputed genotypes is not as large as the variance of the fully observed genotype.
It's basically the input score. It's the proportion of the total variance that's correctly imputed.
Yeah. So it would be 1/2 of the, in this case, the variance of the imputed paternal genotype would be 1/2. And also the correlation of the imputed, actually that's not true. The covariance of the imputed with the observed is actually the same as the covariance of the true with the observed, but the variance is the only thing that differs.
So that would also mean the R-squared would not be
So, yeah, you don't get the same sampling distribution of the parameter estimates as the fully observed. You don't have as much power as if you have fully observed all the genotypes. But in theory, at least, you get consistent and unbiased estimates with valid sampling errors. Yeah?
Sorry, is it possible, or is there any point to like using other observables to help the imputation along, like observables about the parents?
So you mean like if we had the parental phenotypes or something? Yeah, I mean, in theory, what you're doing here is if you wanted to do full maximum likelihood kind of inference with missing data, you would want to use like an EM algorithm, and you could include the, if you had the parental phenotype, you could include the parental phenotype, and that might help you impute. For an individual SNP, this kind of thing is really not going to make any difference, I don't think. But if you're at the level of a polygenic index that explains a substantial amount of variation in the phenotype, having parental phenotype information could help you impute the missing parental polygenic index, especially if you use a sort of EM-type algorithm to do full maximum likelihood inference. But I think for at the level of the individual SNP, because for complex human traits, individual SNPs explain so little variation in general, those kind of things are generally not very informative. So you do gain. So I showed you the sibling difference thing before, and on the y-axis here I'm showing the gain and effective sample size, the relative effective sample size from doing the imputation relative to doing the sibling difference thing. So you can gain, if you do this, the imputation using phase data, you can gain up to 1/3 in effective sample size compared to the sibling difference thing. So that's kind of cool. It's like increasing your sample size by 1/3, just doing a statistical method. You don't have to do any more data collection. And actually, for the estimating the non-transmitted coefficient, you can increase your effective sample size up to 50% relative to not doing the imputation. So what's an application of this? So this is from the paper that's in review at the minute, and we've just used the UK Biobank, which wasn't explicitly designed to collect relatives, but has a lot of relatives in it. And based on about 39,000 individuals from this UKB white British subsample, which is a sort of homogeneous subsample of the UKB, reflecting people that self-identify as white British to some degree. So it shouldn't, it's not a diverse sample. It shouldn't have a lot of stratification in it. And I've also controlled for principal components and stuff, so to make it comparable to standard GWAS. And then what I'm showing here on the y-axis is the correlation between the genome-wide correlation between the direct effects you estimate and the population effects you'd estimate from standard GWAS. So this is telling you how correlated are the effects you're getting from the family-based method versus the standard method. And actually, most of the traits is pretty high correlation. So that's sort of comforting that GWAS is not giving completely wrong answers. I guess it depends what you mean by wrong as well sometimes. But like high HDL cholesterol, FEV1, that's a lung function measure. BMI, they're all pretty close to 1. Correlation. Neuroticism looks like it's maybe a bit lower than 1. But then educational attainment and cognitive ability stand out as having a correlation that is, I would describe it as not high. Low is a strong word. It's certainly low compared to perhaps expectations. So it's around 0.75 for education and only around 0.5 for cognitive ability. The cognitive ability measure in UKB is like not very good, so I wouldn't take that as the best possible analysis you could do of IQ or something like that. And also, I think there might be something going on with ascertainment here because only a subset of the UKB took the cognitive ability test. Educational attainment clearly is below 1, and there could be all sorts of reasons for that. But this is just showing that there is some clear difference in what you're estimating from the family-based GWAS compared to the standard GWAS, and that difference could be potentially quite important. And one of the ways that can be important is, in these genetic correlations that Raymond was talking about, estimating this LD score regression, and I can see evidence that at least for cognitive ability and educational attainment that some of these genetic correlations are probably inflated by either indirect effects or confounding factors, assortative mating, whatever. So my opinion on LD score regression is a great method, but it's applied to the wrong summary statistics. It should be applied to the direct effects summary statistics, because that actually conforms to the assumptions of LD score regression pretty well. And yeah, so this is showing, so it's a bunch of different traits, these points, and I'm correlating standard GWAS summary statistics for a bunch of different traits, correlating them with cognitive ability population effects, as you'd get from standard GWAS on the x-axis, and then the cognitive ability direct effects on the y-axis. And because these points are like going towards 0 from the diagonal line, they're going towards 0, that's showing that in general the correlations with the direct effects are lower, and actually some of them are basically just 0 when you use the direct effects. So clearly some of what's contributing to apparent correlations between traits, genetic correlations, is not direct effects, at least.
Pardon. Why isn't the brown dot, which is cognitive ability, why isn't that on the 45-degree line?
Well, that's because the correlation, so this should be 1 because I'm taking population effects from one sample and I'm comparing them to population effects in another sample. So this should be 1, basically, and it is. But then I'm taking the population effects from one sample and comparing it to the direct effects, and it's below 1, and that's actually concordant with this. This result. Actually, the estimate is a bit higher. It's a slightly different analysis, but similar. Because the direct effects of cognitive ability are not perfectly correlated with the population effects, and that's because there's some sort of confounding in these summary statistics that's different. It's not perfectly correlated with the direct effects, whatever this confounding is. Indirect effects, population stratification, assortative mating. There's something, collider bias, ascertainment. I don't know. And then I also looked for evidence of residual stratification. So I was saying before, I think principal components are not really perfectly suited for adjusting for recent structure, principal components based on common variance. So this is showing evidence for stratification that persists after principal component adjustment. And what I'm showing is the correlation of summary statistics for traits with genetic associations with birth coordinate this is the east axis and this is the north axis, and those associations are adjusted for principal components, and the associations with traits are adjusted for principal components. So this is really what's existing after adjustment for principal components. And for the direct effects, they're basically clustered around 0. So the direct effects are not really structured in the population that much. Actually, one exception is educational attainment, where there does seem to be some evidence for difference between north and south, which kind of supports the Abdeloy analysis I criticized before, although it's smaller in the direct effects than the population effects. If you look at the population effects, they line up along this northwest to southeast axis. So we see that even after principal component adjustment, these standard GWAS summary statistics are correlated with the sort of genetic structure, the geographic structure in the population, and they seem to be lying on this axis, northwest to southeast. Probably a lot of it is driven by London in the southeast versus not London. And the correlation, London, people have a higher income, socioeconomic status in London versus the rest of the country, the northwest, like Manchester, whatever. And there's some genetic structure there that's correlated with the socioeconomic status, and these traits also have a correlation with socioeconomic status, and that's why they lie on that axis, or at least that's my hypothesis for it. And we can see that that's been driven more by the non-transmitted coefficients. So like BMI is a really, the non-transmitted coefficient is pretty significant. So is correlation with the geographic structure. So is height, even though for the direct effects, height is like exactly 0. So even for height in the UKB, white British, relatively homogeneous sample, there's evidence of this geographic stratification that persists after principal component adjustment. All right, so that was the GWAS component. And now I'm going to talk about variance components and polygenic indices, so trying to get the GWAS bits, like this individual SNP. This is more about trying to get the overall genome-wide bits. And going back to the original model, well, actually, I should maybe, does anyone ask questions about all of that before I go on? All right. So going back to the original model, this is the component coming from the direct causal effects. This is this environmental confounding component. And I'm going to ask you to imagine performing a joint regression onto all of the SNPs, the individual's phenotype onto all of the genotypes of all of their SNPs, and also the parental genotypes of all of their SNPs. So at each SNP, you're controlling for the parental genotype in this big joint regression. Then the outcome of this regression could be written as this, where you have the direct causal component is unchanged, and then we've basically, so actually this residual is not the same as this residual, I should say, but basically we've split this environmental confound whatever component into this component that's correlated with the parental genotype, so all the SNPs and then some residual component. So that allows us to talk about the component of the environment that's correlated with genotype and then the residual component, and then this is the component coming from the direct effects of things, of alleles that you've inherited. And from this way of writing the phenotype, you can derive a decomposition of the variance into these three components plus the residual. And this VG is the first component, and I'm just going to assume random mating for now because it makes things easier. The VG component, the random mating genetic variance, that's just the variance explained by all of the, and I'm assuming the causal variants are on different, we've got independently segregating sites as well, that's why you can just sum it up like that. So the random mating genetic variance is just the variance explained by all the direct effects across the genome. Then there's this VE tilde G, and I wrote it like that because it's kind of like you're regressing the environmental component on the parental genotypes, on the genotypes. That's the variance of the environmental component that's correlated with parental genotype. So this is capturing the gene-environment correlation in some sense. How much variance is captured by the bits of the environment that's correlated with the genotypes. Yeah?
Why does the eta have an L subscript?
Oh, that's just a mistake from an earlier talk where I used L's instead of K's. Sorry for that. And that's also a mistake from that. The L should be K's. And then this component is the variance due to covariance between genetic and environmental factors. So you can quite generally decompose the variance as this. So you've got the variance due to the causal genetic factors, the variance due to the component of the environment that's correlated with the parental genotypes, and then the variance due to the covariance between genetic and environmental factors, and then the variance of the residual component that's uncorrelated with the genetics. And we can also define this RGE, like a gene-environment correlation thing, although it's not exactly correlation with the entire environmental and residual component. It's really the correlation between this component and that component. And that will come in handy for when we're thinking about what polygenic indices might be picking up when we fit them in different family-based models.
Excuse me.
Yeah.
I'm having some trouble distinguishing C from V. Is that V? Because there's like in the regression equation, there's two components.
Yeah.
And then the variance equation is 3.
Yeah.
Where do they come from?
So this is the variance of that.
Okay.
This is the variance of that, and this is the the variance due to covariance. So twice the covariance of these two. Yeah. Yeah.
Sorry, you're still assuming random mating here?
So yes, I am. Yeah.
Because if not, I mean, these are just capturing maybe some sorting effects, right?
So when you have non-random mating, things become more complicated for this. So you can still write, you can write down an equation, you can write down the trait like this, but what these, so I should point out I wrote eta here to differentiate them from alpha before, because alpha was like the marginal when you're doing it marginally, just one snip at a time. This is like a joint thing. So yes, these etas could be capturing non-causal things to do with the non-random mating. That's certainly true. For the purposes of thinking, it's kind of easier in a way to think of it as a random mating population and these etas being like indirect effects. That makes a lot of math and in the thinking easier, but certainly when it comes to the real world, it's not that simple, and what these etas are capturing can be very complicated. It can be the stratification, or it can be the correlation with other genetic factors due to assortative mating. Yeah.
So my question is regarding the potential non-linearity, especially when we look at like educational attainment.
Yeah.
Like especially if you look at like direct and indirect effects, you would assume they might be more pronounced at early stages. So if we are just assuming like just linear, then would that really maybe hide the effect? Because early on, I'm not sure like how like the distribution of like EAA is like, if it's sort of truncated, or if we, for instance, like define it in a slightly different way of like saying, okay, below elementary or high school completion or things like that, would that make a difference, especially?
Yeah, I mean, yeah, no, I think you're right in that depending on how the phenotype is measured and defined, a linear model might be more or less appropriate, especially when at the level of an individual SNP effect, a linear model is usually okay because the SNP effects are so small that the linear approximation is kind of all right, although again, the whole issue of like gene environment, if you're looking at interactions and things, it's more complicated. But certainly the level of just doing finding a GWAS coefficient, a linear model, even on a binary outcome is kind of okay. It's maybe not the ideal thing to do, but it's computationally a lot simpler, so that's why it's often done. But yeah, I mean, you could think if you had a case controller or a binary outcome, you could think of this as the underlying linear model that you then put through a transform like a logit or probit or whatever transform that would then give you the actual observations. That would be the model for the actual observations, and this would be the underlying like liability threshold or something like that. So often things can be put back into a linear model through some kind of transformation. So it's kind of the fundamental way to think about it, I think. But then when it comes to practical issues, yeah, like the linear model is not always appropriate.
I think going back to some of Dan's lectures, right? Like we generally we assume that things are flexible and we allow for nonlinearities and whatever crazy things you can imagine. And then you can imagine projecting that into the linear space. And what that means is that the coefficients here are going to have some sort of interpretation of what's kind of the average effect. So even if that SNP has a different effect in different parts of the distribution or people in different environments, the coefficients here are going to represent kind of what's the average effect of the SNP, across these environments.
Yeah.
It's a bit subtle, a bit trickier than that even, but I think it's a reasonable intuition.
Yeah. I mean, I think in statistics, you're always estimating some kind of an average effect. But I mean, yeah, whether you want to use a nonlinear model or not, I think when you're fitting if you're fitting variance components and stuff, that's maybe more tricky issue than just doing an individual SNP analysis, because you don't have this linear approximation kind of thing. Doesn't work as well sometimes. Although people do actually still fit these Greml models to binary outcomes and whatever, and it kind of works okay. So yeah. Okay. So then we're going to think about how these different variance components might contribute to covariances between pairs of individuals, between relatives or whatever. And just a reminder, I guess I already talked about this, but at the level of a sort of chromosome, IBD is when two individuals share a segment of DNA from a recent common ancestor, such as a parent, and this is like a sibling from a single parent. These siblings are IBD, say it's the father, the IBD in this segment from the father, and this segment from the father, this segment they're not IBD, and you can think about measuring relatedness based on the fraction of a genome that they're sharing IBD. And this is kind of the basis of a lot of the more traditional ways of estimating heritability, like twins, that you're assuming, MZ twins have a relatedness coefficient of 1, and that relatedness coefficient is defined in terms of IBD segment sharing. And you can think about more general relatives rather than just relatives defined based on some pedigree relation or zygosity. You can think about more general relatives in terms of like what fractions of the genome they share IBD. And the basic assumption here is that when you want to move to more general relatives and actually, well, just in general for IBD-based methods of estimating heritability and understanding phenotypic covariance in terms of genetic relatedness, is you're assuming that the genotypes are the same between two individuals when they're inherited from the same common ancestor in an IBD segment. So that's just a haploid thing, actually it's not a diploid thing. And you assume that they're independent when they're not identical by descent. Actually, when you have a finite, this isn't exactly true, and you can, anyway, that's the technical aside. But basically, the covariance between a pair of individuals' genotypes is given by the variance of the genotype times the relatedness coefficient between the pair of individuals. And this relatedness coefficient is twice the kinship coefficient. And the kinship coefficient is the probability that if you select an allele at random from one individual, it's IBD with an allele selected at random from the other individual. And this can be broken out into, so the kinship coefficient is 1/4 times the sum. This sum is the sum of the proportion of the genome that they share IBD when you look at the paternally inherited genome of one individual and the paternally inherited genome of another individual. So that these four, if you want to compare two individuals, you can compare the paternal inherited part of one to the paternally inherited part of the other. You can compare that to the maternally inherited part of the other person. Then you have the maternally inherited part of this person compared to the paternally inherited part of the other person, and the maternally inherited part of that person compared to the maternally inherited part of the other person. So you have these four pairs across the four possible parent pairs, and the fathers, the paternally inherited bits might have these bits shared IBD, the maternally inherited bits might have this bit shared IBD, and you'd be sort of adding up all the fraction, what fraction over these four pairs is in these IBD segments. That's giving you the kinship coefficient, the probability that you select an allele from one individual's IBD with another individual, and then you're taking the average over the four of these pairs, and then you need to times that by 2 because you have a diploid genotype, basically, and that gives you the relatedness coefficient, which is 1 for MZ twins, 0.5 in expectation for DZ twins, etc.
I have a question about the IBD-related kinship. So when we do IBS kinship, like we expect it to be somewhere between 0 and 1 usually, but sometimes it goes outside. Are these going to be strictly between 0 and 1 because it's about like literally how much IBD sharing is? Or
no, so the maximum is actually 2.
Oh, well, 0 and 2 then, right?
Yeah. So so in an infinite outbred random mating population, it should be between 0 and 1. When you have inbreeding, it can be between 0 and 2. When you have finite ancestral population size, it can actually be negative.
Okay, so this does have that kind of funny
Potentially, yeah.
Rounded features. Okay.
So, this idea of like relatedness, the covariance between individual genotypes being proportional to the relatedness coefficient, which is a genome-wide thing, that's the kind of assumption underlying all these methods, traditional methods of estimating heritability, which I think can sort of be summarized and all thought of together as operating in this space that has phenotypic correlation on the y-axis and relatedness on the x-axis. Yeah?
What's the standard deviation or variance on the relatedness across different groups of people?
I mean, that's a complicated question. So MZ twins, I guess, don't vary at all, for example. So DZ twins and siblings vary quite a bit, and they've got the most variation, at least if you don't consider strange inbred relative classes like that don't exist in most populations. But if it's sticking to the sort of normal like relative pairs, siblings have the most variation in relatedness, and I think it's around 0.04 is the standard deviation. So I'll show that in a second actually. Then the variation goes down as you go down the relatedness spectrum. So the mean and the variance are related basically until you get to MZ twins, and it's the highest. There's no variation there, I guess. So the ACE estimator in twin studies, that's basically the gradient of this blue line here. So all these different methods can sort of be thought of at a high level as fitting some kind of linear relationship to points in this space that has phenotypic similarity on the y-axis and genetic similarity on the x-axis. And the key assumption that it's making is there's no greater environmental similarity to MZ over DZ twins. This animation is not working as it's supposed to. These Gremel methods, I'll say more about them later. That's what estimates SNP, heritability in unrelated individuals. That's looking at how phenotypic similarity changes with relatedness for unrelated individuals. Can still be biased by indirect effects and population stratification. This methods I call Kinship method, which was used to estimate heritability of IQ. There's a lot of media attention in 2018, and I think this method is not the best because basically just looks at how this similarity changes with relatedness across all these relative pair classes, and it has no real principled way of separating genes from environment. There's this method called SIB regression. Well, I call it SIB regression. I think Peter Vischer himself calls it full SIB regression. I don't know is that different a name, but yeah, from 2006, which is super old now, and that's showing this variation that you're asking about in relatedness for sibling pairs. So it's kind of like this normal distribution around 0.5, at least in outbred populations. And what this method does is it looks at how phenotypic similarity changes with relatedness within sibling pairs. And because the sibling pair relatedness variation is generated by the random segregations that occur during meiosis, this is a very robust way of estimating heritability. It can be biased by indirect effects from between siblings as well, actually. They're just a real nuisance. But the problem with this method is it needs a huge number of sibling pairs to have much power. So while I was at Decode, I developed this method that kind of generalizes the SIB regression thing to all relative pair classes. Basically, the idea is it's just like with the individual genotype, the individual SNP, you're controlling for the parent's genotype and using the random variation around that parental expectation. You can do the same at the level of relatedness. You can look at the relatedness between a pair of individuals, and you can compute the expected relatedness between that pair of individuals on the basis of the relatedness coefficient between the parents of one individual and the parents of another individual. And the expected relatedness between a pair of individuals given their parents, the relatedness between their parent pairs is just 1/2, and that's this diagonal expectation line. So this is this related, based on IBD, the relatedness coefficient between the parents, and then the relatedness between the offspring. And so this is showing the siblings, like for the SIB regression thing. And then this is going down, so you can see there's a lot more variation here. than there is here, and the variation goes down. But most pairs in a population are distantly related. So most of the density is down here, and that's where most of the power comes from. But you're basically using this variation around the expectation given the parents again, but at the level of relatedness to try and estimate heritability in a way that's not biased by environmental confounding. And that was the basis of this paper, where I took advantage of the decode data where there's like 55,000 people with both parents' genotypes, which I think is a lot more than any other data set, apart from maybe some of the commercial ancestry kind of data sets, 23andMe and Ancestry.com and whatever. So we're able to use this method I call Relatedness Disequilibrium regression, which uses this random segregation, and it is connected to that variance decomposition that I showed you before, in that basically the covariance between individuals due to direct effects is given by R, their relatedness, so this is like the relatedness matrix. Then the covariance between individuals due to this environmental component is correlated with parental genotype, and that would include indirect effects and stratification and stuff. That's given by this parental relatedness coefficient. And then the covariance between individuals due to covariance between genetic and environmental factors is given by this slightly more mysterious relatedness matrix, which is like the relatedness between the offspring of one individual and the parents of another, and vice versa, the average of those two. And by fitting this covariance model, you can get what should be an unbiased estimate of the heritability, with some caveats about assortative mating that I'll talk about in a sec. But just to compare, so RDR is what we call it, to compare that to this kinship method, which doesn't really control for the parents. It does some modeling of shared environment between siblings. You get these huge differences for height, educational attainment, BMI, age of first child in women, and clearly there's some sort of bias in that kind of method where you're not controlling for shared environment properly. If we compare so Iceland is such a small population, there aren't enough twins to actually do a proper twin study. So compared to Swedish twin studies, so the caveat here is not the same sample, but the twin estimates are much higher on average than what you get with RDR. And RDR, because it's using the IBD segment relatedness, it should be getting something very close to the full heritability, although it might miss some variance due to very rare variance. That's probably not true for the SIB regression thing. So this paper that I released also had the biggest ever sample to use for the SIB regression method. And the SIB regression uses siblings, so they really share like everything other than de novo mutations not shared by siblings. And you still see that on average the heritability is lower than what you're estimating from twins, although again the caveat is it's a different sample. And then we can compare to Gremel methods. So I'm just going to give a brief introduction to Gremel. So Gremel is, so there's notation, I'm using L's here again, but that's okay because the L is there too. So basically you're assuming some kind of standardized genotypes, and in this model it's a little bit different because they're standardized to have variance 1. And you're basically looking at measuring relatedness using genome-wide SNPs, and you're looking at how phenotypic similarity changes with relatedness measured from genome-wide SNPs for pairs that are basically unrelated. They're close to unrelated, as in they don't have any recent like common ancestry. Well, that's the idea at least. And it's different from RDR, as I presented it before, that uses IBD segments. It's using SNPs, and it's modeling the SNP effects as random effects. And if you model the SNP effects as random effects in this way, you get this marginal distribution for the phenotype vector y in terms of this relatedness matrix based on SNPs, which is basically just so the relatedness between a pair of individuals is kind of just like a genome-wide correlation between the SNP genotypes, and a residual component, and then the Gremel SNP, and you're doing this in unrelated pairs of individuals. That's the idea. So you avoid shared environmental effects shared between like siblings, for example. And the idea is you estimate this parameter and divide that by this sigma squared g, which gives you the variance explained by the sort of effects population effects, I would say, of the, it's related to this additive, this is basically SNP heritability as defined before. And I think a useful point is there's a difference between estimating relatedness with SNPs and with IBD segments. IBD segments is like more like what we traditionally thought of as relatedness in the pre-GWAS era. It has to do with shared ancestry. SNP relatedness is really more like a genome-wide SNP similarity metric. And the two become very similar for highly related people. And I think I remember proving once that if the genome is of infinite length, the SNP relatedness would become the same as the IBD relatedness. But with infinitely many independently segregating sites, essentially, infinite number of chromosomes. But it's not. So the SNP thing actually varies a lot more than the IBD thing. The IBD thing is like the expectation of the SNP thing. And the SNP thing varies around the expectation given the IBD. So you can see that people with no shared IBD segments, they still have quite a lot of variation in their SNP relatedness. But they become kind of the same, or very close to the same, highly correlated for, these are siblings, for more closely related people. But the SNP relatedness thing is really using this variation in SNP similarity around, that even exists for sort of unrelated individuals. But that's why it's only estimating the SNP heritability, not the full heritability, because it's the IBD segments are capturing everything apart from very recent mutations that occurred since the last common ancestor, whereas the SNP thing is just the normal LD between SNPs. So then you can ask, so what would Gremel pick up if you have indirect genetic effects? So we can do a simulation where the variance explained by the direct genetic effects is 20%, the heritability is 20%, and then if we have a index, that's an old term that I don't really like anymore, but indirect genetic effect from parents 1/3 the size of the direct effects that are perfectly correlated, then you can say the total variance explained by direct and indirect genetic effects of transmitted alleles is 35.56% approximately, and Gremel in GCTA, that's sort of one of the standard pieces of software used for estimating. He's doing restricted maximum likelihood in general. It can do any sort of REML kind of inference. It doesn't have to just be the Gremel model. Estimates basically this total variance explained, and that can be expressed in terms of these variance components. And actually, in the computer exercise, there's basically a simulated trait that's quite similar to this. Whereas if you can do a SNP version of RDR. There's kind of it's like Gremel, except you're adding the parental relationist matrices estimated from SNPs. That'll just estimate the component from the direct effects. So yeah, the standard sort of SNP heritability, my point here, the takeaway point is the standard SNP heritability, it's including variance from indirect genetic effects and covariance between direct and indirect genetic effects, potentially also stratification. So it's not really the same heritability, in my opinion, as traditionally estimated and defined in like the behavior genetics literature. It's more like a R squared you get from an infinite sample size from the regression or something, rather than heritability as traditionally thought of.
So do you simulate with the covariance? Is that why it's a...
So the direct and the indirect effects are perfectly correlated here.
They're perfectly correlated.
Yeah.
So what is it in the simulation? The total heritability is supposed to be
20% for it's a heritability. If you define heritability as variance due to direct effects, it's 20%.
It's 20%. And then how much and then you're saying a third of the direct effects. So a third of the 20 are supplemental.
So this equation works because they're perfectly correlated. So the total variance explained by the combined direct and indirect effects of the transmitted alleles is 1 plus 1/3 squared times that 20%. So that's the inflation. If the correlation is lower, then it would be less.
So so there's inflation. Okay.
It's
due to the inclusion of direct effects are correlated.
Even if they were uncorrelated, it would still be inflated. It'd just be inflated less. Yeah.
So you've got that inflating the heritability, but then of course you only simulated causal variants that were already on the that were already on the chip, right? Like
Yeah.
Like so, because you were talking about before this whole thing about how you miss the rare variants when you are using unrelated people because you're not because they basically don't share the haplotypes.
Yes.
So then you've got two competing forces basically.
So this is ignoring all of that complexity. This is just a simulation where we know the causal SNPs and we're estimating relatedness matrices with the causal SNPs. So we're ignoring the complexity of when the causal SNPs are not in the relatedness matrix, used to compute the relatedness matrix. So yeah, you can't just directly compare Gremel using SNPs and unrelated to the results I showed before. But this is what this slide is trying to do the comparison more fairly, because I'm using a SNP version of RDR where you compute the relatedness matrices using SNPs, which has similar properties to using IVD segments, and comparing that to a version. I'm not using REML for the inference because of the structure of the Icelandic data. You can't really construct a big sample of unrelated individuals because it's such a small population. So I'm just using unrelated. It's more like a HE regression, this using unrelated individuals. And I do see a fairly big difference for educational attainment and age of first child in women, somewhat big difference for height. And it seems plausible to me that part of the difference for educational attainment and age of first child in women is driven by stratification indirect effects. Height, probably just assortative mating. As you know, this is a slide based on there's a nice new paper from Richard Border, who's now at UCLA, but he was a PhD student of Matt Keller, where they look at how assortative mating biases different estimates of heritability. So this is an important caveat when you're comparing different ways of estimating heritability, that they can be affected by assortative mating in different ways, and in potentially quite complicated ways, annoyingly. And a result from this paper that still kind of like blows my mind really is that Gremel will estimate. Oh, I should probably say what all of this is first. So if VG is the variance due to direct effects in a random mating population, if you add assortative mating and you let it go to equilibrium, so after a certain number of generations, assortative mating increases the variance. After a certain number of generations, it goes to equilibrium. And under certain assumptions, you can show that equilibrium value is equal to the random mating value divided by 1 minus the correlation between the spouse's genetic components, or the mother and maternal and paternal genetic components. And also that will induce a change in the heritability at equilibrium compared to in a random mating population. Gremel apparently will, in an infinite sample size, estimate the random mating one. So assuming we're at equilibrium, you apply Gremel, infinite sample size, it'll estimate the random mating thing, but in finite sample size, it converges super slowly to that. There's some random matrix theory that shows that. LD score regression has it overestimates the equilibrium heritability, and then RDR, SIB regression, and the twin ACE model will all underestimate heritability under assortative mating by the same amount, basically, because they're using this within family segregation variance to some degree to estimate the heritability, and because chromosomes and whatever segregate independently in a family, that removes the inflation due to assortative mating, so they will tend to estimate, RDR and SIB regression will estimate the random mating variance, and then if you turn that into heritability, they will, under certain assumptions, they will underestimate the equilibrium heritability, and they all do it by the same amount. So assortative mating can't explain the differences between like RDR and twin estimates, but it could explain some of the difference between RDR and LD score regression and Gremel estimates. Yeah?
So from the reading list, the Evans et al. Paper in 2018 talks about how the inflated heritability could be due to Gremel being highly sensitive to assumptions. So is that part of?
So yeah, I mean, Gremel doesn't do any modeling of assortative mating. I think exactly understanding how assortative mating affects Gremel estimates is pretty complicated. So I would recommend this paper if you're interested. It's kind of it's not an intuitive result to me, this anyway.
Yeah, I think that Luke Evans paper, I think the big distinction between the models that they're comparing are what they assume about the distribution of effect sizes across the minor allele spectrum.
Yeah.
That I can remember.
Yeah, so I think the original Gremel method didn't do any kind of partitioning of SNPs, and I think that made it more liable to being biased because of this strong assumption it makes that all SNPs, it essentially makes the assumption that all SNPs contribute the same amount to the phenotypic variance. And I think the more recent versions of Gremel, which are worth knowing about, to be fair, I don't have any slides on it, like Gremel LDMS, it stratifies things by minor allele frequency and LD score, and in doing that, I think it makes it more robust to this violation of model assumptions. Well, it's a more complicated model, so I guess the violation is less as well of the assumptions. But yeah, I mean, I think estimating things from SNPs requires stronger assumptions, I think, in general, than using IVD segments is a point worth making too. All right, so now this PGI analysis in families. So that's wrong. That should be WK squared. Apologies for that. Oh, no, it shouldn't. That's right. Oh, yes, right. I only made these slides this morning, so... So we're going to imagine a general polygenic index, which is just some weighted sum of the SNPs. And these weights can be anything. So it's a linear function of the genotypes. And it's normalized to have variance 1, so that you have this 1 over root v, where v is the variance of this term. So that we've normalized the PGI to have variance 1, which is standard practice, what people do. And then we can think about doing various things with a polygenic index. And often people just do standard regression, phenotype onto polygenic index. But that could obviously be biased by stratification and things like that, just like the individual SNP case. So just like the individual SNP case, we can take into account the fact that given the parental genotypes, parental alleles, the variation in the offspring polygenic index is due to these random segregations in a family, so that's independent of confounds. And just like in the individual SNP case, we can take the expectation of the polygenic index given the genotypes of the parents, and that's just the average of the father and the mother's polygenic index. So again, you can split an individual's polygenic index into the component, the expectation given the parents, the average of the paternal and maternal PGI. It works because it's just a linear function of genotypes. So it's a lot of the intuition and theory for single SNPs carries over. Not all of it, but a lot of it. And I mean, the single SNP case is really just a special case of this, where WK is 1 for one SNP and 0 for everything else. And so we can split it into an average expected component and a random component. And then, just as before, we can do this sort of family-based regression, where we're regressing onto the individual's PGI and the parent's PGI, where the parent's PGI is the sum of the paternal and maternal PGIs. And we can ask, what are these coefficients if we do this regression? And you can show in general that these are the coefficients, a somewhat complicated expression. And I should also point out that this is assuming this normalization, random mating, and independently segregating variance. So this actually doesn't apply in the case of assortative mating and whatever, which I'll talk about in a sec. Under these assumptions, you can show that these are the coefficients you get in a regression where you're controlling for the parent's PGI. And depending on what weights you put in, those coefficients are going to reflect different things. So if you use the truth, so that assuming no noise obviously deflates things, but assuming no noise, if you use the direct effects as the coefficients, then you would get the square root of the genetic variance would be the coefficient on the direct on the individual's PGI, and then the coefficient on the parent's PGI would be reflecting gene-environment correlation and the variance of the environmental component correlated with parental genotype, which could include indirect genetic effects and stratification factors. If you use the non-transmitted coefficients instead, it's kind of like the opposite of this, where the delta coefficient reflects the gene-environment correlation, and the eta coefficient reflects the environmental variance correlated with parental genotype. And this is the typical case, so this is something that no one really does yet, but maybe they should. But this is a typical case where using a PGI constructed from standard GWAS summary statistics, so population effect. And in this case, your coefficients are kind of like complicated. So the delta coefficient's reflecting the direct effects, but it's also reflecting gene-environment correlation. And the eta coefficient is going to be reflecting the environmental component correlated with the parental genotype, including indirect effects. It can also be reflecting the gene-environment correlation too. So it's just a caveat to say that, you know, if you're doing these family-based analyses with polygenic indices, exactly how to interpret these coefficients is not trivial, and it depends a bit on what you're putting in as the coefficients, or is what you're going to get out. And obviously, adding noise complicates this, and also non-random mating complicates this, such as assortative mating. And this is kind of a subtle but important point. So, and just to explain this, I'm just going to say, imagine that you have a PGI constructed from one chromosome. Under random mating, that PGI is uncorrelated with the whole rest of the genetic component of the trait, because chromosomes are segregating independently. In random mating, there's no correlation across chromosomes. However, under assortative mating, the PGI on one chromosome is going to become correlated with the rest of the genetic component of the trait on the rest of the chromosomes, because assortative mating generates correlations between causal alleles on different chromosomes. So if you would then imagine doing this family-based regression, where you regress on the individual and the parent's PGI, the direct effect, the coefficient on the individual's PGI, that's going to reflect the effect of the PGI from that chromosome only, because chromosomes segregate independently within a family. So that correlation that exists in the population due to assortative mating goes away. However, it will load, this correlation with the rest of the component of the trait will load onto the coefficient on the parent's PGI. So if you do an analysis with individual and parent's PGI, and you see a significant coefficient on the parent's PGI, you can't interpret that directly. Sorry, I shouldn't use the term directly again. You can't interpret that simply as evidence for indirect effect or stratification. It could be assortative mating. It could be that that's reflecting this correlation between the PGI and the genetic component of the trait that would be orthogonal, uncorrelated with the PGI under random mating, but becomes correlated with it due to assortative mating or population structure. Okay.
So can I ask, just pragmatically, this is really interesting. So if you do your within SIB GWAS in a discovery sample and then construct polygenic scores for just the direct effect and use it in a prediction sample,
Yeah.
in which you don't have parental genotypes available, it sounds like you're still getting some bias associated with the correlation between the direct and the indirect. Or another way of putting it is that you would get a higher R squared than if you did that same thing in a bunch of adoptees.
Yeah, so if there's gene environment correlation, like through indirect effects with the family environment, then yeah, if you just regress on a PGI constructed from direct effects, it's not going to give you an unbiased estimate of the direct effects because of the gene environment correlation. Yeah.
But if you had a sample of adoptees that were in some way representative of the broader population in terms of their family environment, then and genotype, then it would give you an unbiased estimate?
So if you have a population where there's no gene-environment correlation, then, and there's no assortative mating, then yes. However, even with no gene-environment correlation, if there's assortative mating, the coefficient on the parents bit, if you just do the standard regression, that's going to reflect correlation with the remaining component that would be orthogonal to it on random mating. I mean, I guess you can argue whether that's bias or not. In some sense, it's maybe not if it's just direct effects.
And then if you have the parental genotypes in the prediction sample, does that change things?
Well, yeah. So if you imagine a situation where there's just direct effects and assortative mating, and you construct a PGI from direct effects, then you would estimate, but it was an incomplete PGI in that it didn't explain all of the variation, then the eta coefficient would still be significant because it would be reflecting this correlation induced by assortative mating. However, if the PGI was complete, it captured the full component, then there would be, the eta coefficient would go to 0. So it's partially a function of incomplete data in a way. Like if you had perfect estimates of the full genetic component, including rare variants and stuff, then this assortative mating thing would go away. Yeah.
Just looking at your population effect, like if we didn't really care about getting the delta PGI estimate to be the right thing, but we're interested in what the relative size of delta PGI versus eta PGI. We could like take the difference, because there's this confounding CGE term.
Yeah.
The difference out if we took the difference between those two, right?
So you mean the difference between these two?
Yeah. That still is going to give you sort of
that would tell you, I guess, whether VG is bigger than VE tilde G.
Yeah.
But that's under assumption of, that's assuming there's none of this assortative mating bias, which, there definitely is.
The PGIs are potentially differently powered, right?
Yes. So this is assuming no noise.
Right.
And the R squared you get from the direct effect PGI would in general be different from the non-transmitted PGI, which would in general be different from the population effect PGI. And like if you fit also, depending on whether on this RGE term, in theory, you could get a higher R squared by using the direct effect PGI for the individual and the non-transmitted PGI for the parents, but that wouldn't necessarily give you an unbiased estimate of the direct effect of the PGI unless you had a perfectly correct model and estimates.
I mean, I guess it seems like there's this draw to just like compare the direct and the indirect the R squareds from each of them, and then to label one as more important than the other. But I guess my worry is that the R squared is proportional both to the heritability and the amount of measurement error in the PGI.
Yeah.
And if there's differential measurement error, we may be trying to interpret as differences in heritability accounted for by direct versus indirect effects, but it could really just be differences in the amount of measurement error.
Well, yeah, and you also have the correlation that's going to play into the correlation between the non-transmitted and the direct effect components. So I think using PGIs to make statements about variance components, I think is tricky, personally, compared to using more direct methods like Gremel or RDR or whatever, which can directly estimate variance components. But I think you can at least interpret them as capturing part of these variance components under certain conditions. But the sort of mating thing, especially for like EA, if you're using the EA PGI where the sort of mating is really strong, that makes this interpretation is kind of alright, but in the real world it's not that simple either, because of this issue, which is definitely real and important. And this. So when I was at Decode, I was sort of still, I made some contribution to this paper, mainly the title, but But this paper did kind of have a big influence. And the main sort of result, I think, that shocked everyone, and this is kind of just doing this regression, basically, was if you calculate the R-squared based just on this delta coefficient in the PGI, if you use EA PGI, it's around 1/2 of the normal R-squared. And that observation has been replicated in lots of other data sets, and actually in the UK, it's only around 1/4, so it's an even bigger difference. So this difference in R-squared you get, or like the delta coefficient being much smaller than the delta plus eta, and this delta coefficient being smaller than much smaller than delta plus eta, or delta and eta being of comparable magnitude. That's quite replicable, but what the interpretation is still up for debate. In this paper, we argued it was parental indirect genetic effects, and we did some adjustment for this assortative mating thing, but it was a little bit. It relied on some quite strong assumptions, that adjustment, and I think the jury is still out on the relative contribution of like assortative mating, population stratification, indirect genetic effects. But this idea that the PGI is behaving differently within families in the general population, and the coefficient is much smaller when you control for parental genotypes. That's a very robust result, and it's clearly important, but we don't exactly know why. We haven't given a complete accounting of all the factors that explain that. But if you're doing PGI analyses, it's important to remember that, I think. And just a note to say, you can also use imputed parental PGIs, and there's this functionality in the SNIPR package for computing parental PGIs from imputed parental genotypes and doing this kind of family-based regression with the imputed parental PGIs. And just an example of an application of this in the UK Biobank. So most of the time, most of the sample in the UK Biobank, we don't have the parental genotypes. But by doing the imputation, you can actually fit this more complicated model where you have indirect sibling effect and the non-transmitted parental coefficient. So you fit the direct effect, the indirect sibling effect, and the parental non-transmitted coefficient. And when we did that, we see the indirect effect from the sibling come out to basically 0 for education. This is for an educational attainment polygenic index on educational attainment. We saw no evidence for an indirect sibling effect, which is in contradiction to what Dalton said last week. But he was using DZ twins, so I'm kind of interested in whether there's some difference between general siblings and twins in this respect, or just curious whether that result replicates in general, and whether this result replicates, I guess. But again, you can see that, so this green bar is the delta coefficient from before, and the blue bar is the eta coefficient, and in the UK they're about the same magnitude. So it's interesting to think what's going on there, because if we go back to this, the R-squared would be only 1/4 compared to 1/2 in Iceland. So clearly something different is going on in the UK to in Iceland. And just to finish, there's this computer exercise that I'm going to put on the Dropbox. And it's basically the simulated phenotype with direct and indirect genetic effects from parents. It shows you can compute the relatedness matrices for the RDR model, and you can estimate that model using REML and GCTA, and you can compare that to the estimate from the GREML model in GCTA. You can impute the missing parental genotypes using SNIPR, and you can estimate direct, and in this case they are indirect genetic effects, using the imputed parental genotypes. So if anyone's interested, it'll show you how to use all those different GCTA and the SNIPR software package and do estimate these different models. So yeah, thank you for bearing with me through all of that. My back hurts, it's been so long. So.